So the differentiation of these T cells is super high yield. This might be one of the higher yield things in the immunology section because there's so much stuff. It's pretty dense. So let's just simplify how to think about it. So of course things in the thymus undergo positive and negative selection, and we'll talk about that. Positive selection, the first thing that happens, well, it happens on the outside of the thymus. It happens in the cortex. So what you do there is you say, well, are you able to bind? Is the cell able to bind? Well, yeah, fantastic. Okay, good. So now you can progress to negative selection. If, of course, the cell fails its positive selection round, it gets destroyed. If the cell then goes into the medulla, it's the more inner part, so as you progress inwards, you go towards the medulla, that's where you have negative selection. Okay, well, how tightly do you bind to self-antigens? Oh, you bite really tight? Oh, that's no good. You get destroyed. Okay. Well, what if you don't bind too hard? Oh, great, then you get a stay. And here they talk about AIRE, autoimmune regulator. So this thing's actually pretty important because it helps show and present and make material that these T cells end up getting exposed to. So let me pause it for a second and pull up a good image. Yeah, here's a pretty good image. So of course we're in the medulla. This is your regulator protein. Okay. Well, what is it making? Well, it's making a bunch of, self-antigens, you can call it. It gets presented to the T cells. Does it bind too hard? Yes. Negative selection failed, so boom. But if you don't have this autoimmune regulator protein, what happens? Well, then you don't going through negative selection. So now you can understand how you can have a whole bunch of T cells that haven't been vetted, and they're going to start attacking, your own body's antigens. And there you can, of course, imagine how you can have a litany of autoimmune complications arising from that. All right, so let's kind of go through this stuff. One of the big things that the exam's going to test you on, and assume you know, so it's one of those things that's going to be an easy point for you, is asking you about MHC1, MHC2, is it CD4, CD8, and then asking you about Th1, Th2, and asking you is it CD4, CD8. And that's not just a guess. So anyway, these are the big helper cells that you're going to think about. Of course, they're all CD4 positive. So you can kind of see how everything sort of influences everything else, right? So Th1 secretes interferon gamma, upregulates itself. Also secretes interleukin 2, which is huge for Th2. That's how you can remember it. 2, 2. Okay? And as is tradition, Th2 is going to secrete interleukin 4, which helps upregulate itself. Okay? But at the same time, remember how Th1 also secretes interferon gamma and interleukin 2, right here. Well, interferon gamma downregulates this, but interleukin 2 upregulates it. So the actions of T helper cell 2 it's not just by itself, it's going to play off, well, how does Th1 interact with it? But similarly, Th2 also makes interleukin 4, which downregulates Th1. So you can kind of see how things are positively and negatively interacting with each other. Okay. Now these T regulatory cells are also influenced by Th1 because it makes interleukin 2, sorry. However, these T regulatory cells end up getting downregulated by Th2. Which is funny because you always think of T regulator as the one downregulating everything. But when you look at a lot of stuff, Th1 in my mind is the one that's like kind of getting everything started. But Th2 is really doing the heavy hitting, So I have a pretty bad drawing here about kind of how everything influences everything. Now, this is very basic, of course, and I'm missing a lot of, um, interleukins, and it's not clear what these arrows are talking about. But hopefully you can see how a lot of these things just upregulate itself and are dependent on each other. So it's a very complicated system. It's not something that's immediately intuitive, but you'll start seeing, just how complicated things get. Big thing, of course, for each activates macrophages and cytotoxic T cells. Interferon gamma. That'll do it for you. TGF beta. Big thing in autoimmunity. And we'll see more of this interleukin 17 as we go along. And of course, right here, you've got your eosinophils being activated here. What else are we missing? You'll notice how cytotoxic T cells act similarly to natural killer cells, killing with perforin and granzymes. You also see how, in a similar way to natural killer cells, where they look for MHC1. Cytotoxic T cells are looking for MHC1 on virus-infected cells. So I got a question for you. Is this an endogenous or an exogenous thing? Well, remember, endogenous, cytotoxic. And then let's look at this thing right here, FOXP3. FOXP3 is the big thing in all of this. We already talked about regulatory T cells and how it prevents autoimmunity. TGF beta is a big part of that. So anyway, FOXP3. Well, think of it as the primary regulator for the regulatory T cells. Similar to the AIRE, you're going to end up having problems with your autoimmunity if you get rid of it. So what's the big thing to understand here? Well, hopefully you can see how all these things play with each other and play against each other, and kind of the intricate, complicated, but beautifully nuanced dance that goes on between them. Positive and negative selection, where it occurs, and what it means. Okay. And then AIRE and FOXP3. That was a long one, but thanks for sticking along.